Hello class, today we will learn about the phylums Zygomycota and Ascomycota, and we'll also study a bit about extracellular enzymes. From the phylum Zygomycota, we will study Rhizopus and Phycomyces blasculianum, and from the phylum Ascomycota, we'll study some powdery mildews such as Ancinula salicis and Plumeria graminis, and we will study as well nectar canker caused by Nectocina varina, ergot disease caused by Claviceps purpurea, white rot caused by Sclerotinia sclerotirum, black knot disease caused by Apiosporina morbosa, and we'll study as well sodium mold and witch broom caused by Eposporina collinsi. So our first microorganisms is Rhizopus, and it causes soft rot of different fruits. This is a very common post-harvest disease, and in the fruits, the infected areas are water soaked and very soft, and usually we get an outgrowth of hyphas, which covers the affected portion and produces Sporangiophoros and Sporangia. This is a saprophytic fungus which has non-septate hypha. In these pictures, we can see the hypha outgrowth and the Sporangiophorus, which are these pinhead blackish heads. Rhizopus has two different types of reproduction. The first one is the asexual reproduction, which is through sporangiospores, which are one-celled and non-motile spores, and they are produced within an sporangium. Here we have the picture. First of all, we're going through the diagram. Here we have an sporangiophore, and on it an sporangium is produced, and inside of this sporangium the sporangiospores are produced as well. The sporangium has different parts, so we have here the rhizoids, the sporangiophore, apophysis, which is the base that keeps the sporangium together, the columella, which is the ones responsible for feeding the spores, and the spores. Different sporangiophores are usually joined together through an estolon, as we can see in this picture. This is an actual photograph of one slide of Rhizopus, and we can see here this is an sporangium which is full of sporangiospores. We have here the sporangium wall, which is the outer part of the sporangiophora, the columella, which is the one that keeps it together, the apophysis, and here the sporangiophora. In this one, we can see only the rhizoids and the estolon, which keeps together different sporangiophoras. Here we have an dazed sporangium. Here we don't have the sporangium full of sporangiospores. They're already out. And we have here the columella and the collar. The sexual reproduction of Rhizopus occurs through zygospores, which are the sexual spores of this fungus. These are the overwintering or resting stage of the fungus. The formation of the zygospores have different stages. In the first one, we have a hypha of the negative mating type and one from the positive mating type. And they come together and a gametangia is produced, then it continues to grow and a cell wall is produced around it. Then a zygote, which is 2M, is produced, and finally we get the zygospores. The parts of this we have in this photograph. The gametangia, when they come together, then we get the zygospore here. They're black and very thick wall, and the structure that keeps them, that sustains them, are the suspensors, as we can see here. Our next microorganisms is Phycomyces blasculianus, and this one is a saprophytic fungus, and it is heterothallic. What does it mean? It means that we need to have positive mating type strain and one negative mating type strain growing together so we can get the zygospores formation. In this plate that we can see here, we have two different microorganisms growing. The first one is the positive mating type in the left part of the plate and the negative mating type in the right hand. And this line of brownish structures in the middle is where the zygospores are formed. So we have in this fungus as well two different kinds of reproduction. The asexual reproduction through sporangiosperms as well, and the sexual reproduction through zygospores. Here we can see how the zygospores are developed. In the first phase, we get the progametangia and they come together. Afterwards, this black cell wall forms. The gametangia develops. The suspensors are the one responsible of keeping the gametangia together, and then they keep to evolve, and dichotomously branched appendages develop, and at the end we get the developed zygospore, which is held by the suspensor, and it has dichotomously branched appendages. Now we'll switch to the phylum Ascomycota. And first we're going to review, because we have already studied the powdery mildews. In the first powdery mildew we have is Anseinula salicis, and this fungus as well has two different kinds of reproduction. At first we have the sexual reproduction, which is through ascospores, which are produced within an ascus, and those asci are within a fruiting body, which is the Cleistocesium. In the case of Anseinula, it has coiled appendages. And it is important to know in this fungus that usually most of the fungus thallus is outside the leaf. In this picture we have a section of a leaf, and here we have the thallus of the fungus, and right over the leaf are the Cleistothecia. Here we have the photograph of a cross-section of the Cleistothecium, and here we have the ascus, where the ascospores are in numbers of 8, and we have the coiled appendages, and this is a complete Cleistothecium. The other powdery mildew we're studying is the powdery mildew of cereals and grasses, and it is caused by Plumeria graminis, whose anamorph or the asexual name of the fungus is Oidium monoleoides. The asexual reproduction of this fungi occurs through conidia, and the sexual reproduction through ascospores as well. Again, the fruiting body of powdery mildews are the Cleistothecia, and in the case of Plumeria, we have Cleistothecium with meseloid appendages. In this picture, we have the asexual reproduction. So we have different conidiophores which produce on top of the leaves, and we have a chain of different conidia. The conidia which is closest to the conidiophore is the youngest conidium in the chain, and the one that we get farther apart is the oldest conidium in the chain. Here we have the Cleistothecium of Plumeria, and we get to see it which it has meseloid appendages. This is an obligate parasite, and therefore it produces Austorium, which are for this fungus bipolar and digitated. They are very hard to see with the microscope, so here that's the reason why we have a picture of them here. So we have their bipolar, so they have these two digitated forms in each side of the Austorium. Our next fungi belonging to the phylum Ascomycota is Nectocina varina, and this pathogen causes the nectar canker. The endomorph or sexual name of this fungus is Tubercularia vulgaris, and this pathogen causes canker on many tree species. the asexual reproduction of this fungus, as well in all the Ascomycota fungus, is through ascospores, but in this case the fruiting body of the microorganisms is different. The ascospores are produced within a perithecia, which for this fungus is red, globose, and is produced in groups. In this picture here, we can see the reddish perithecia, and the asexual reproduction of this fungus is through conidium, which are produced within another fruiting body, which is an Sporodocia, which is long, stipulate, and disc-like. So the sexual stage of this fungus is reddish, as we can see in the picture over here, while the anamorph or the asexual is, or its kind of skin color, orangey, as we can see right here or right here. But how does perithecia and Sporodocia look like? Here we have a perithecia. It's a pear-like structure where the ascospores are produced, and a Sporodocia is sort of like a glass of wine, a martini glass, where the conidia are produced on top. Here we have the bark, the host tissue. Now we're going to study a very interesting pathogen, which is Claviceps purpurea, and this pathogen causes the gut disease of cereals and grasses. This pathogen, the symptoms are developed in two different stages. In the first stage, we have the honeydew stage, where creamy droplets of sticky liquid go out of the grains, as we can see here, are exudated. And in the second stage, sclerotia are produced and replace the grains of the plant. It is important to know that this pathogen is poisonous to humans and animals. That's why it's very restricted, and whenever we're growing wheat or barley, they check for this pathogen. The sclerotia of this pathogen are the overwintering structures, and it has two different layers. The first one is the medulla, which is composed by branch and loose hyaline hypha, and the rhinth, which is the outermost structure part of the sclerotia, which is the rhinth, and it is a pigmented wall which provides resistance of the sclerotia towards degradation. Once the sclerotium germinates, it produces between 1 and 60 stipes, and this is how it looks when it germinates. So we have here a germinating sclerotium where different stipes are produced, as we can see, and on top of there we have the stromatal heads. How does a stromatal head looks like when we made a cross section? We can see here how it looks. So here is the stromatal head, and inside of it we have different perithecium, which are pear-like structures where the aci are produced. And in each one of those perithecium, an 8 long global multicellular ascospores are produced. So remember, the ascospores are the sexual structures for the reproduction of the pathogen. Now we'll study Sclerotinia scleroterium, which causes white rot of canola, and this pathogen causes different symptoms. Among them we have the shredding of the stem, as we can see here, stem bleaching, you can see in all of these pictures, and also sclerotia of the fungus are produced usually inside of the plant stem. The sclerotia are this kind of blackish structure which look like small soil particles, but they're not. They're the fungus structures. Again, for this pathogen, the sclerotia are the overwintering stages, and whenever they germinate, they produce the fruiting bodies, which are the apothecia, which are small. They are thin stalks which end in a cup-like structure, and the cup of the apothecium is lined up with aci. Here we have the germinating sclerotium, here are these types, and here the apothecia, which are cup-like. This is how a cross-section of an apothecium of Sclerotinia sclerotherium looks like. But which are the parts of it? So first, we have the hymenium, which is this part, and it is covered with the aci and paraphysis. Then we have the subhymenium, which is right here, which is a compact zone of hypha with thin wall cells. Then we have the ectoxipulus, which are hypha cells which are short, polyhedral with a very thin wall. And finally, we have the medullary excipulum, which are long hypha cells with very thin wall. But if we go and look in more detail, this is a section. This is a close-up of one of the parts of the apothecium right here. So here we have the paraphysis, which is this kind of this structure which doesn't have nuclei. Then we have the ascus, we have the ascospores, we have the subhymenium, which is the part of the apothecium which is below the hymenium, which is where all the aci are located. Our next fungus is Apiosperina morbosa, which is the cause of black knot disease. I'm sure that if you go to River Valley, you're going to be able to observe these symptoms, which are rough black areas, which are known as the knot, which kills the infested part of the tree. The knot is the overwintering stage of this fungus, and it is capable of producing numerous pseudothecia. This is a cross section of a gall or the knot that we saw in the slide before, and on top of it we have this Nomenum pseudothecium, and inside of the pseudothecium we have the aci and the ascospores lined up, as we can see in this photograph. Apiosperina collinsi is the cause of black leaf and witch's broom of Saskatchewan berries. What are the symptoms this pathogen cause? We have the edges of infected leaves roll downwards, and the underside of the leaves, as we can see in this photograph, are covered with black mycelium and different pseudothecia. Also, the fungus is capable of penetrating the shoot tips and stimulate the growth of many new shoots, causing enhanced branches, which is what we know as witch's broom. Here we have this photograph of how witch's broom looks like in Saskatchewan berries. And finally, as you can see, we've gone through different fungi, and we've seen that they produce overwinter structures, which are the sclerotia. These are the overwinter structures, and when they germinate, they usually produce different kinds of fruiting bodies. And it's very important to note that the sclerotia can be of different shapes and sizes. Here, for example, we have a sclerotia from Thifula, here from Sclerotinia borealis, here from Sclerotium ralphsi, from Sclerotinia sclerotiarum, and we can see that they have different sizes and shapes, and we have these ones of Thifula, which are very small, and this one from Polyparius, which are huge. The last part of this lab are the extracellular enzymes, and one of those are the polyphenol oxidase. These enzymes are responsible of degrade the lignin, and therefore they're very, very necessary for the pathogenicity of different fungus, among them Rhizoctonia solani. In this lab, we have a video of how a PPO test is performed using a cabbage medium. What happened with this medium? We prepare a medium, which is this purple one, a liquid medium, from cabbage and then put the fungus to grow inside it. When the fungus starts growing inside, it begins to use the different compounds of the medium, and among them we have some phenols in the cabbage extract. And when the fungus starts degrading it and producing polyphenol oxidase, there is a change in the color of the medium towards yellow, and it occurs because the fungus produces a yellow subproduct whenever it degrades these polyphenols from the cabbage medium. So now that you have learned the basics of our lab, I invite you to go to the link on eClass and complete the third quiz and use for that all the information on the slides, the videos, and lab guide. And don't forget that you will be able to complete the quiz until 1 hour before our next lab, and that you can go back and forth as many times as you want. And please don't forget to submit your lab once you're done, and enjoy it. Bye.